our next speaker is my colleague, Daniel Feliciano here at Janelia. So yeah, I will summarize. I think it basically will tell us how AI helps us to provide the organelle insight of a cell state.
Yes. All right. Thank you, everyone, and thank you to the organizers for allowing me to present today. So today I'm going to tell you about some of the work that we're doing in the lab, trying to bridge both the tissue and the organelle scale using a technique that we are developing in the lab called Spatial Organellomics. So this was inspired by our interest in understanding how cells within tissues are capable of sensing their local environments and how they are also capable of adapting if there is a change in this environment. Within tissues, we have different cues, including hormones, nutrients, and oxygen. And together, these cues can affect cell biology by changing cell fate determination, differentiation state, and cell bioenergetics. Together, these changes allows organs to acquire their functional heterogeneity, which is very, very important for these organs to activate different pathways like regeneration pathways or disease resistance. While most of the characterizations of cells have been done at the transcriptional level, recent work has shown that organelle architecture provides with information about the cellular state. And this is because organelle architecture is the consequence of both transcriptional regulation and protein regulation. So this new perspective underlines the potential of imaging organelles as a powerful alternative to study cellular diversities in tissues. Most of the information that we have in terms of organelle adaptive responses, we have learned from tissue culture cells. And we have learned that organelles are capable of changing their morphology, their position within the cell, and their numbers. This allows cells to adapt to different environments and allow them to acquire different metabolic states. However, due to the complexity of imaging different organelles and segmenting these organelles in the tissue context, very little is known about organelle adaptive responses in the context of physiological environments. This is why there is a need of more imaging approaches to quantify organelle architectures within tissues and organs. So in the lab, in collaboration with the Funke Lab, we have developed a new workflow for 3D imaging segmentation and for measuring of organelle features. We have used the liver, as Jonathan mentioned, because it's the best model system for us, due to the gradients that are formed in the liver, where hepatocytes are exposed to different set of cues, which allow these cells to acquire different functions. And this is known as metabolic liver zonation. So despite liver zonation has been already described for about maybe 100 years, very little is known about how cells within the liver regulate their organelle adaptive responses and how the structural heterogeneity within the different zones are regulated. So for this, we are imaging very large regions of the liver using confocal imaging, very simple approach. And with this, we make sure that we are imaging all three zones. We also image different organelles, including peroxisomes, mitochondria, and lipid droplets. And we use phalloidin to label the cortical actin, so we have the boundaries of the cell. So we have developed a new Python package called LipZones that allows segmentation of hepatocytes and the organelles. And with this, we can quantify about 100 different organelle features at the single cell level. Because we have the information about the cell position, we can go back to the data and look at every single organelle readout for every cell and see how they are diverging within the tissue. If we take all these organelles information and put it in PCAs, or principal component analysis, and apply Gaussian mixture models, we can categorize these hepatocytes in 5 different categories with distinct mitochondrial, peroxisomal, and lipid droplet characteristics. Sorry. So we believe that these 5 different categories represent hepatocytes with different functions. And as I mentioned, in liver zonation, depending on where the cells are localized, is where you acquire the function. So we believe that these categories were localized in the same place within the hepatic acinus. Surprisingly, we saw that that is not the case. These different categories are spread out all across the acinus in differential frequency proportions. And in fact, they are in each one of these zones in different proportions. This suggests a model of liver zonation where zones in the liver are made of a combination of structurally distinct hepatocytes coexistent in different proportions, which is very different from the current model. We were very excited for this result, so we decided to validate it using a different data set, and we use a spatial proteomic data set from Matthias Mann lab, which in line with our results also show 5 different hepatocyte categories with different proteomes. They didn't analyze the way the hepatocytes were organized in the hepatic acinus, so we repurposed the data and apply our spatial heterogeneity analysis, and we found striking parallels. So together we can say that hepatocyte heterogeneity is not only determined by position along the periportal-pericentral axis as previously thought. So we also wanted to know how these organelle architectures are influenced by changes in the nutritional conditions of these animals. So we used 2 other models. We used the extended fasting model and the Western diet model. So in both of these conditions, we see a complete remodeling of the organelle architecture that was accompanied by changes in zonation of the organelle features. While in the fasting state, there were some of the features that remained zonated the same way as the control. In the Western diet fed mice, most of the different features actually shift their zonation status. This suggests that hepatocytes use different organelle adaptation strategies during nutritional challenges. We used PCA to look at the different hepatocytes from these 3 groups, and we saw that they segregate in different clusters. And we did subclusters with GMM and found that in both fasting and Western diet, the number of categories decreased. In fasting states, we saw 4 categories, while in the Western diet, we found 2. In total, we have 11 distinct categories if we add up the 5 categories from the control samples. So we look at the distribution of these new categories across the hepatic acinus, and we found that in both cases, in fasting and Western diet, the hepatocyte heterogeneity profiles were reduced. And this was reflected by both the proportion graphs here and the Pittsburgh heterogeneity indexes. This allows us to conclude that nutritional challenges reduce hepatocyte heterogeneity by decreasing the organelle signature diversity. So together, we hypothesized with these results that organelle signatures encode information that links subcellular architecture to metabolic states, and we can harness this information to use machine learning to try to predict different cellular states. So to test that, we use MLP models using organelle signatures and the features as descriptors. And we tried to train 2 models to make 2 different tasks. One was to determine between the 11 different hepatocyte categories, and the other task was to differentiate between the 3 different nutritional conditions. We were very excited to see that in both cases, our predictions were very high accuracy, and these demonstrate the power of organelle signatures for predictions. So encouraged by these results, we decided to test this in the context of progression of liver disease. So we use a model for early liver disease where we fed animals for up to 2 months for Western diet, and we collected samples at different time points. We apply our S-organellomic workflow, and fed MLP model with this data, allowing this model to predict between these different stages of the disease with an 80% average accuracy. So together, organelle signatures represent a structural fingerprint that not only allow us to differentiate between cellular status, but also allow us to understand better the status of the tissue. But like any other technique, it's not perfect. It has some limitations, and one of the limitations is that is because this is using diffraction imaging microscopy, so we cannot see a very important aspect of organelle dynamics, which is the organelle contact sites. So to solve this, we joined forces with CellMap, a project team here at Janelia, to image the full hepatic acinus using FIPSIN. This is a gigantic volume composing 12 million cubic microns. It has about 2,000 cells, so we use this volume to segment every single cell, and we segment 7 different parts of the cells, including the boundaries, the nuclei, peroxisomes, mitochondria, the mitochondria inner membrane, lipid droplets, and the endoplasmic reticulum. We also quantify the 6 different contact sites, and with this we acquire information about the organelle interactome in the liver, allowing us to quantify the amount of these different organelle contact sites and their position across the hepatic acinus. We also characterize these to understand their differences. And since this data had organelle contact site information, we wanted to test whether this information increased our resolution to detect different hepatocyte categories. So doing so, we apply our S-organellomics strategy, and we found 7 different categories this time. And similar to the light microscopy data, the different categories were spread out across the acinus in different proportions. So the goal is like everyone want to generate a multi-scale model where we can have all the information, in this case, structural information of liver zonation, to understand hepatocyte heterogeneity in the different zones. It would be great if during this symposium, many collaborations can arise so we can incorporate different modalities to these models to make them more robust. So I want to thank everyone involved in this project. I want to thank you all for your attention, and I'm happy to answer any questions. Thank you.
Thank you.
Hi, Daniel. Amazing talk. Thank you.
Thank you.
I have a clarifying question. For the last plot, you showed a UMAP with 7 classes. Is that combined with the features you extracted before, or is just based on the interactions?
Everything. The
Everything
interaction
combined.
plus the organelles on their own and the different features of the organelles.
Very cool. Thank you.
Very beautiful pictures. I'm just curious, in the fasting condition, it seems that there are more mitochondria, like more higher density, and also even more lipid droplet. Could you elaborate a bit more what is going on in the liver?
In terms of the mitochondria, because this is a nutritional scarcity, what you expect is to see a mitochondria biogenesis, because mitochondria is the powerhouse of the cell. So if you're lacking nutrients, you want to have more mitochondria. In the case of the lipid droplets, because this is an animal, obviously the starvation response or the fasting response will make the adipocytes release lipids, and the liver absorb of these lipids. So that's what you see in these droplets.
As described, I think it's maybe this audience also curious. For the model training is great for livers. So do you think how easy to translate them to other organs? Yeah.
That's a very good question, and I don't know at this point, but in collaboration with the Spinoza Medina lab, we're trying to apply the same strategy to pancreas to see if it works the same way.
That'd be cool. Thank you.